Good evening and welcome to everybody. My name is Carolina Rojas. I'm a research associate at Roatan Marine Park, and I will be moderating today's webinar on coral spawning monitoring and assisted sexual fertilization. This webinar will be given by one of our research coordinators, Damaris Dueñas. She's been working with coral spawning monitoring since last address coral reefs by implementing coral seeding methods and technologies. So today she'll teach us how it's going to be done. At the end of the webinar, there will be a section of questions and answers where Damaris will be able to answer all of your doubts and questions. As well, you can leave your comments on our chat. And with nothing more to add, I'll leave you with Damaris.
Thank you, Carolina, again, for introducing into this webinar, and thank you everyone for joining on time. This webinar, as Carolina mentioned, is what we're going to do. It's basically what we are planning to do this year in our efforts that are pivoting a bit along into coral spawning and coral rearing, rearing baby corals and as sexual reproduction technique. And we've been given this webinar already in Spanish, and for today we would like for a lot of most of our diver communities, dive owners, dive managers as well that are living here at Roatan. We can introduce to them what our efforts will be and how they can join as well. And this is mostly like a training as well for every one of you that are already joining this webinar. And just to mention, the webinar will be recorded as well. And we have it on our YouTube channel. So we'll begin recording. And now, yeah. So let me share my screen. Just give me a moment so I can start presentation. All right, just to make sure everyone, can the presentation be seen on the screen, Carolina? Yeah.
Yeah, it looks good.
Cool. All right. Well, thank you again. And I will introduce today's webinar, telling you a bit more telling you a bit more about what are our plans for this year. So mostly this will be a lot about coral spawning, and one of our first objectives is to validate our 2022 coral spawning calendar, and I'll explain a little bit more about what is this calendar about and how is it done. And we'll focus on 10 priority species because we have lots of species of mostly stony corals that we are giving priority to after our devastating disease, stony coral tissue loss disease. This is like one of our strategies that we would like to go into our coral restoration now. We will also conduct gamete fertilization and larval rearing for one species. And I will tell you a little bit more, and this is due to one of our biggest collaborations this year with Secret International, that we're collaborating with them for 5 years. So each year we will be having developing our capacities and sharpening our skills into this technique, and trying to increase our species for fertilizing them. And for this year, we will begin with one species that I will also like comment to you along the presentation. Also about ensembling cribs. Cribs is like a pretty name that they use. It's a Secret technology. I will talk about it later as well. It's sort of like pools, and those are like baby cribs as well that we were ensemble and put corals in the water and let them grow or settle until they're a little bit enough grown to put it back to the reef and outplant those baby corals. We will also, and we're still planning to collaborate together with local partners as we have done previously, because this is one of the strengths that are and make possible projects like this, involving all the local community. And another type of involvement will be, we will develop this Ocean Youth program that it's basically focusing educational and trainings like this into the local students, like young adults here at Roatan, and that we will take on as well, provide them not only in water experience, but also conservation and marine biology first knowledge to them, so they can later further careers in this area. And moving forward, I just want to begin with a brief introduction of what we'll talk about, and is about the life cycle and a little bit of the life cycle and reproduction of corals, coral spawning prediction, coral spawning monitoring, gamete collection, assisted fertilization of corals, rearing of larvae in the scripts, which are called coral rearing in situ basins of this SECOR International organization. Just a preview as well of why are corals really important. Well, as we all know, they provide shore protection from waves and hurricanes. They also are habitats, food and shelter, and they provide food and shelter to marine wildkind, and provide also resources such as human food security and protection. So this is really a broad of what the reef provides to the organisms itself and human community. So going further a bit more of coral reproduction, we know that the corals are living creatures or organisms that they, as long as any other organisms, they are born, they grow as adults, and they, once they're mature, they reproduce. And what happens, these creatures or these organisms are really amazing because they have two types of ways that they reproduce. They can reproduce through sexually through spawning, or using different type of reproduction, which is called through fragments, which is asexual. So they don't need this connection of the female male genetic variability, and they simply, once their fragments or these colonies are, because of hurricanes, or because of weather conditions or damage, they break down these fragments and stay or mobilize or anywhere in the water, and they can become a new colony out of them. So we can see, for instance, in this image, when storms and hurricanes, they also provide this type of opportunities for corals to move along the reef, because once they're established in a single place, they don't move, right? But this is another type of evolutionary traits that they have adapted through a long history life for them to be successful to where they are now. And so they are able to reproduce through spawning and through fragments. And for those types of reproduction is how conservation has also studied them and has a lot of information on reproducing them in fragments. And now we will see what's all of the sciencey base as well that has been done to now understand a little bit more and trying to reproduce them sexually or through their spawning. Is going along the life cycle of coral. This is an elkhorn coral. So once they're mature, they can so their fragmentation, if that's a way that they reproduce, but they can also spawn once they're mature. So in this massive spawning, all those different colonies around an area, the eggs, once if they're not siblings around, they can fertilize, and once they fertilize, they go along all these phases, which they begin into this first larvae stages, which is called planula, when is their only time when they're mobile and they're trying and moving in the water, trying to settle down. And once they settle down, this little polyp stays in a single place and it stays forever in this place and just grows into a juvenile. All those stages are very critical for all colonies, corals, because this provides them the opportunity to increase their populations. And not only their populations, but also there's two ways of reproducing. Also, not only increases like the structure of the reefs and itself, the population of the colonies, but their resilience through variation of genetic variations. So through spawning, so we'll talk about a little bit of what's that reproduction through spawning. In fact, the corals are so incredibly interesting that whenever they spawn, there are two types of ways that they spawn, or there are different types of colonies that they spawn. One way of them is like, there's some species that they have female or male, and that means that they can, whenever they spawn, they can spawn or either sperm or gametes, or female gametes. What does this mean? For instance, maize and pellet corals, if we have two different colonies, one in front of the other or next to each other, and one of them will spawn this year, let's say, sperm, and the other one will also spawn sperm, this means that there were two male colonies, and probably in a different site, there will be another colony that will be spawning female gametes. And this is when we also say that these are really critical and very important types of reproduction. And mostly when there's a lot of diseases, we are very likely to see that their reproduction rate decreases as well. And this is when all these conservation projects come along and try to assist them into complete their fertilization. So maize collars, spirit corals are really, well, basically in danger itself because of this like very low rate into reproduction. So they have female, male, and there's another type of reproduction that are called hermaphrodites. And these ones are like this brain corals, like star corals, elkhorn, stockhorn corals, that they produce itself the same colony, female and male gametes. What does this mean? That either there could be like two colonies, like they're not siblings, two colonies just right next to each other, and they can fertilize if they spawn, either be because they both produce female or male. So it doesn't matter, like in the other example that I told you, it was if there were two female or two male colonies right next to each other, there was not the possibility to fertilization to occur. So either strategy that both colonies have is trying to evolve into what their adaptation has been throughout their environment. So they have these two types of reproduction. And this is also one of the ways it's different whenever we monitor them. And this is, I'm going to talk a little bit further as well, how we monitor. And because we have different stages after monitoring, we will collect their gametes. So we'll be different collecting sperm, because sperm is like nothing, you see nothing, to just see like a little bit of kind of like even liquid, and we are just diving in more liquid, right? So it's really interesting, like the type of tools that we're going to use. Let's go to the next slide. All right, so I was talking about predictions. So coral spawning, it's basically once a year, the corals provided by signals through the moon cycle and water temperatures, and also synchronizing through sunset time. All of those factors combined provide them hints for hormonal hints that provoke or develops the spawning in each of the species. So whenever we say like once a year, is one of those months, or from those months that are the hottest months. So there's corals here in the Caribbean, they spawn mostly from July to September, October. And then the moon cycle, so every species has a day after full moons, because they synchronize with the full moons. So days after full moons could be from 1 to 9 days or to 13 days after full moon. Each species as well is synchronized to release like hormonal cues for them to spawn. And this is why also we can predict that, because it's basically what is happening is that the whole environment is providing this perfect cues for them to spawn, right? And if we know what months, oops, hello, can you hear me? All right, so I'm going to shut down my video because I think the internet connection is not working really well. So if we know already what months and what days after the full moon, and it depends as well the sunset times. Most of corals, they spawn after sunset times. And what does this mean? There's a lot of scientific research being done throughout the years that people go and monitor, simply trying to see any spawning. So some of people have already predictive times. It can be like 1 hour, 2 hours after sunset. And let's say, for instance, the sunset is at 6. So it will be monitoring at 8 during the night, or 9 or 10. Because it's really predictive, we can create these calendars that I was mentioning. Here on the next slide here. So here's one of those calendars, and there's a lot of people and organizations working on this spawning predictions calendar, because one of the main objectives of this is creating this information for managers to increase or help to manage marine protected areas, as well as these species, to get to know them even better. And also because of a lot of conservation actions and restoration programs, they're trying to add this into their restoration techniques. So this is how one of those predictive coral spawning calendars look like, and this is for Eleuthera. This was from 2020, and as you can see, it tells by month, and every single colony or the species is on the left, the second column, and here are the scientific names, right? And after that, it says a window time, which is minutes after the sunset time, and after the months, it says as well every calendar day, and then days after full moon. And you can see that in the month here where I have this red box is where we call it like the peak months, which a lot of or more colonies add into their biological calendar to spawn. So August, September, and October are those like peak months. So once we know the peak months, once we know all about like predictive cues or times and dates that are on our area, we can elaborate our own calendar. And this is what we've been doing. This is now this year we can say that we have elaborated like the first corresponding calendar for Roatan, and this includes probably Honduras, because or sunset times, and basically all Honduras has the same time. That has no more, it's not really extensive to vary, right, from one place to the other. So thanks to Secor International, as well as a lot of collaborations that we have with different now doctors that are working with Secor, they have provided us a lot of feedback and giving us direction in how to improve our predictions. And here you can see that our predictions will begin from May, June, August, September, and there's a lot of the rest of cover, but it didn't fit here. But here is, as well as the previous calendar, it tells us months, the colony names, as well as the days. Here, the full moon is represented as this little oval here on the top of the 0. It means days after full moon, and then hours after full moon. Here are the minutes, and the colors, which we can represent here, are basically telling us from light to dark if there's enough data to say yes, there will be spawning that day if you monitor. And this calendar is really valuable information that we will be sharing with everyone and the managers, because we'll also and we're probably one of the pioneers organizations that are working on this. Will provide this information and provide capacity as well for other professionals, Honduran professionals and managers of the MPA, for them to include them as well as one of the things that will provide more information to a proper and continue with the proper management of our corals. So we have elaborated this calendars, which we will further as well when monitoring. We'll provide an assistance to all the divers and us ourselves that will be monitored, will be out in the water trying to validate this calendar. So this is predictive. That means that we need to go into the water now and validate what we have already predicted. And this is what this whole webinar will talk about, and how to do every single stage from monitoring, collecting, rearing or assembling these cribs, and out planting back these corals. So it's a lot of activities, a lot of stages, and how everyone can be involved in each one of those steps. And as I mentioned earlier, we will first months of corals, and we will have to prioritize species. And from those species that we have given priority are from stony corals that have been hit from SCTLD, as I mentioned as well. And we will work with those that are under the red list. Some of them are under the red list as vulnerable, endangered, critically endangered, and are those corals that you can see here. So we have, we're gonna work with 10. I think I'm missing one picture here, but those are the species that we'll try to validate their spawning times this year. And we will read one species that we'll show you later. So monitoring coral spawning. Introducing to you what are this monitoring. We monitored last year here at Roatan several species. Several volunteers that were really committed, and we worked together, and we were in the water like for an hour or more. And it's really something constant, because we need to monitor different colonies. Also, we need to record species, date, and time of spawning, because this is very valuable information that will help us to modify and validate this calendar and future calendars. So also, but it's really shocking for sometimes is that there will be nights when there will be no spawning, but the monitoring will be continuous. So the monitoring is continuing during the week. And it is really important to know that coral spawning will be very useful for coral restoration and future research. So basically, in this whole webinar as well, I will be like telling you as if I was teaching you that you will conduct every single stage as yourself. So this is whenever we do the monitoring. So we collect data, and that really valuable data that we collect is similar to this data sheet that I provided, or that we were provided by Corallium and Seacorp. Those are different organizations that also they monitor along the whole Caribbean, and there's like a standard information that we collect. This data sheet is for one of those species that have female male, as you can see on the left of the first column, and they're called gonochoric species. And this is like one of our great collaborators and volunteers. We have been working together with stony coral tissue loss disease as well. And they were monitoring in a dive site called Happily Ever After. They collected information for a specific monitoring that we were looking for in this monitoring. They were monitoring pillar coral, you can see there, and they place the timing, the monitoring start time, stop time, and if there were spawning or not, and so on and so forth. So this is the monitoring data. And for monitoring planning tips, well, as you can see on the picture as well, torches were essential, and we did it through, or it can be done through night snorkeling or scuba diving. We also will need like a light on the surface as references, either be for divers whenever are down below, and they can have like a surface reference to go back to, and also for avoiding, or for safety and avoiding any other type of in areas so there's a lot of traffic, like boat traffic, and even during the night, those are like safety things as well. And also be present at the site. We need to be present at the site for monitoring 1 hour or at least 30 minutes before and after window time. So in case that you're coming along with us, we're all monitoring together, these are the things that we'll need to have in consideration. So in case that our monitoring or the spawning window of a colony, it's at 7 p.m. So we want to be in the water at least at 6 or 6:30, because remember that this is like a prediction. Sometimes it could be before or later than the spawning time. So another tip for monitoring plan is to select shallow sites that keeps us longer dives in case that we're scuba diving, and provide us the opportunity to be more than 60 minutes underwater, and allows us as well to collect this valuable information after the spawning time. Window. Another and different tip, it would be once we're doing monitoring, is that we tag the colonies. We add underwater markers as well with buoys. So we tag them, as we are looking here at the picture. We assign, once we have the divers, we assign each diver a colony, and we can even assign it up to 2 colonies, which are close colonies. And for species that grow massive, it could be even better just to stay 1 colony per person. So what we also encourage is that we take the camera and the data sheets, and also safety first. So we can see here on the pictures that we were tagging the colonies. This is one of our volunteers that she helped us out even like in the whole process of it. We were measuring, taking information as well for some colonies. And the last picture down below, those were like little markers with buoys. It's like they had weight on the bottom, and they were holding this little lace and floats. So once we were down below and during the night, we have 2 different markers. One is, it was to individualize each colony, which was the tag, and the other one to help us out as reference to just going straight to the colony that we saw during the day. And that's like another thing, that once we go into the water, it's better to recognize a place during the day and to recognize it, to be able to recognize it during the night, because it really changes a lot. Last year, we monitored the species, and it was elkhorn, lobster coral, mountainous star coral, and we monitored it from August, these 2 months, we monitor during September, but these species spawn from August to September, or what has been confirmed in other sites. The elkhorn coral, their days of DAFM means days after full moon, and their spawning days or their windows are from 0 to 6. So from the full moon to 6 days means like a week. And their spawning time, which means the other digits here, which is 140 to 190 meters. AS means after sunset. So that's, in that year, it was at 8:30. So it was like the sunset was at 6, was a 1 hour, 2 hours, 6, 7, 7, yeah, 2 hours, 1 hour and a half later. So we were on the water from 8 to probably 9:30 or 10. And we divided into different groups. One of them were scuba diving, others were snorkeling, or simply, remember Elkhorn coral, we were able to do it snorkeling. So we were snorkeling throughout an hour. And the same you can see in the other species, days after full moon, one of them is 5 to 8. And the mountainous, the same, these 2 star corals are from 5 to 8 days. They're minutes after sunset, the same almost as well. And we were monitoring 2 species in a single dive. So this can happen as well. And it was really useful. And as well, what we said was that 1 species spawned one day, and there was no spawning for the other species until the next day. These are the species that we monitored last year. So counting the the previous slide, plus these other brain corals and this large cup star coral. we saw spawning only from this 2 species, the star coral. And for this year, we will focus on this that I have a red box. This is the groove brain coral. This is the only species also, which is really interesting, that spawns before the sunset. So this will be really easy for us also to introduce diverse peoples or that are beginning to monitor, and because we'll have a little bit more of sunlight, and will help us out also to begin our practices like our fertilization, collecting gametes practices with this species, because it's also the only species that begins their spawning window, their months, like really early, since July or June, we can start monitoring the species. And for the rest of the others, we will definitely monitor or give priority on monitoring on August and September. We're planning to do gamete fertilization with D-Lab, which is, this is the code, like the scientific code for the species, the brain root corals. And further throughout the years, we'll consider these other species as well. So once we have developer skills, for instance, in Dominican Republic, they have been doing this for already 5 years or so. So they're rearing 2 species per year. So they're taking advantage of those long spawning months. So they monitor this groove brain coral and they collect gametes, and they go through all the process until rearing them, which takes up to 2 months or 1 month. And then they go into the next month. Let's say they begin in July, they collect and fertilize their gametes, and then in August, they're ready to collect and fertilize another species. But this means like they have had a lot of trial and errors, and this is what we'll go throughout this year. And we will begin with a single species, and let's see how it goes, this species this year. And then we will think about what other priority and other species we can combine to in a single year, or just 1 year. I'm sorry, 1 species per year. So when we're doing this monitoring, something that I will tell, so everyone who will join or who will conduct or this monitoring is, let's be patient, but then let's dive a lot. That's like one of the greatest recommendations that are given to everyone. So let's be consistent on our monitoring times and patient, because sometimes there will be no spawning. I'll tell you from advance since now, but you'll have a chance to encounter different marine life. And here's some pictures. This is from our last monitoring. The first 2 pictures is here in Roatan, and the last picture is in the Dominican Republic. There were nights, so there were no spawning, but there was still flourishing of life during the night. And we had see different organisms that have different behaviors during the night. But also the magic will appear. And I'll leave you this video. You can see this is a spawning of yeah, so this is a small video, and you can see the spawning in the middle of all the other organisms of this staghorn coral. So the spawning, or the magic as I call here, occurs. So after we have monitored, we have exhaustively monitored for a week, an entire week of the species. They're in their time after the sunset. So the species that will focus on monitoring this year will begin monitoring like 10 minutes or 20, 30 minutes or 1 hour before the sunset, and like 30 or 1 hour after sunset. So once we have collected all this information about the spawning, if we saw or did not see any spawning, for the next month, we'll begin this monitoring probably in May, June, so we can be more accurate with the timing, so we can be ready with the collection of their gametes and fertilization. And so we will have all the skills in monitoring, precise, or already like helping us out throughout or through make sure that this process goes more easily. So the next stage will be ready for gamete collection. And before we do that, we need to have this tool done. We need a gamete collector, and we call them collecting nates. So we're gonna get this materials and we're gonna elaborate them or produce them. So we will learn to elaborate them. And right now we're seeing, like, we're gonna see every single tool that we're gonna use for equipment, and this is what we're gonna use. And later we will create, I'll tell you like how, if you are really interested in working together and in volunteering, etc., we're gonna be creating this equipment, and that will be like our practice sessions. So we'll be creating this nets, and once spawning occur, we'll have to practice as well, like how to place them into corals and how to withdraw them. Because once the spawning occurs, this is what we will be doing. And also we will be removing this collector flakes. So on the picture exactly, it is what it is. This is like, it's almost, this fabric is like a mosquito net, and inside of it, it has some funnels, which helps out for all the gametes that go upwards through the surface, go inside the collectors. And these collectors, as you can see on the last picture, those are like those lab collectors, like sample collectors that are used. We were donated several collectors that are different. Those are like, they look like tubes, but this is the same process that happens. We will learn as well that we will have to cut, we have the measurements, we will cut the whole mesh, but we'll try to stick or attach the funnels as it looks on the other picture with the whole mesh or the whole, to create the net. Then we will attach a lace, a float, and here in the middle, we will attach also using like Coca-Cola, like the little tip of the Coca-Cola, just to make it able to rotate and attach it to the sample collector. And basically this is like really easy to do, and we're gonna learn how to make this, guys. And also not only to make them, but go into water and know how to put them and withdraw them. Those little things that you can see on the base of the collector, those are weights. So we will use small weights, otherwise the whole mesh will just go flying away, and the gametes will not go through the funnel. This is how it looks underwater, replacing nets once we have them. So if you're a really good observer, you can see that there are no weights here. So there's different types of nets as well. This one has a lace. So what it works is like you just make it a little bit tight, because it depends on the species. For instance, this branching coral is really different, and weights can even break the fragments. So for several species, just like there's corals, staghorn corals, we will use this other type of ending for the mesh. And this is once spawning, how it looks. The whole process of the spawning or the gametes going inside the mesh and through the funnel to the collector flakes. So there will be water inside, there will be worms inside, but there will be a lot of gametes. And this is a stage where we're all collecting. And once we have a full like little of this little collectors, what we do is that we remove the lead, and we will learn as well that we patiently remove the lead. And because we're defying gravity, or the gametes are defying gravity, they're not going anywhere else. We're just removing the lead to place another lead on inside, and then closing it and put them all together again in a single container, and putting back on another sample collector, a new sample collector if you want to collect more of these gametes. And once we have all this collection of gametes, and we have all those collectors ready, and we take it and either be on the boat or in, let's say, at a deck, on a dock, or the office, or elsewhere. And so it will look like this. This is the collector full with water, and all the gametes, they will be on the surface as well, the water. So we will conduct the assisted fertilization, which, to sum up, it's simply putting all together what we have collected from the same species into water. So we combine all collected gametes in a container, and we will constantly mix this pool of gametes to resemble the action of waves. This action of waves is what breaks into two or into several pieces the gametes and provokes this fertilization. So for each species as well, there is a salvage fertilization timing. So it's not that right immediately they fertilize. Now they will have to, depending on the species that we will be rearing, we will try to fertilize, we will have to be doing this for 20 minutes as and analyze samples as well, and under the microscope for every 20 minutes after combining gametes. So for some species, it's like 2 hours after mixing the gametes. Another species is like 2.5 hours or less. So it depends on the species as well. So this is basically the whole assisted fertilization, collecting gametes and put them all together and sampling them. Once we are sampling them on the microscope, this is how it looks and how it will be told as well when the fertilization occurs. So we'll be taking samples of those gametes, and once we see it on the microscope, it will look like this. Based on what it has been researched, we want for the whole gametes to be this at 70% of them fertilized. And this is how it looks when they are fertilized. And the arrows, like the red arrows, are pointing into like a split gametes. They look like little buds or berries. So that's when fertilization is occurring. And after this fertilization occurs, and we need to count each and every single one of those little gametes that are fertilized and the ones that are not, so we can take this percentage of fertilization. So when we have reached these X amount of fertilization, we're ready. Either if we want to rear them, we put it into our cribs and our substrates, or what we will also be doing in our first stage is that we fertilize them and put them back into the water. So either way, you're still conducting this fertilization. So you're either, if you don't have the rest of the materials or equipment or facilities to conduct a rearing of this larvae, you simply can stay until this process, assisting in fertilization. And this is the whole technique. So, and you put it back into the ocean, but that's the thing, you just leave it into the hands of the ocean. You cannot either assess or evaluate if those gametes, those fertilized gametes, became into colonies. But you can evaluate like how much of the amount of gametes were able to fertilize, and you can tell that you increase the percentage of the fertilization. So now that we have our exhaustive monitoring, we have conducted also our gamete collection, we have fertilized them. So now we have a pool of fertilized gametes. So what do we do now? So we want to place them into our cribs, or we're gonna rear them into this in situ basins. But before all of this work, there's like previous stages that we need to do before putting them in or using this secure cribs. There's some substrates as well. They're called seating units. And we're going to be using 1,000 substrates in each and every single substrate. And it looks like this, like as it is in the picture, can host several larvae. So this is where the larvae, once it's trying to look for a place to settle, will settle down. So throughout the years, Secor has been developing several techniques and technologies, and they have also experience with concrete seating units or the substrates, but also like made out of ceramic. They will provide us the substrates, and the ones that we will be using are made out of ceramic. And they have found that both of those materials, larvae, they're really happy. I will say like this, like happy, or like this type of surface, so they can encrust. So the ceramic are those like star-like shape substrates, and those are the ones that I will be using. So we'll have to precondition them, and that means like we'll need to locate, once we have those substrates, we'll need to locate them underwater for algal growth, because we want them for like the good algae, like under quotation good, as those calcareous algae that corals like to then settle down into them. So once we have, or we're ready, we're gonna rear those larvae, we have to place the substrates inside some crates underwater for 2 months. So this year we will try to rear larvae in September. That means that since July, we will place down this substrates underwater in a place where there's enough algal growth, but the good algae, like the calcareous algae. So we later use it on September. We use veggie crates. Those are like the ones that you see on the supermarket or market, so we can hold them together and put them together. So once we are going to use these crates, and once we have, we're planning to go into the monitoring and collecting, blah, blah, blah. So we're going to clean the substrates or the seating units days before we use them. Why? Because there's going to be lots of algae, like macroalgae, growing on top of it. Also, if there's a lot of sediment, these guys will be dirty. So we'll need to clean them. We'll need to clean them not really like exhaustively, because we want to leave this like good algae, all the calcareous algae to remain, but remove the other type of algae. Also because the substrates are really good, and other species like to lay eggs on them as well. So in my experience, I remember when I was trying to cleaning the substrates back when I was being trained, there were different types of little eggs found on different like edges that those guys had. So we needed to clean them, but also leave the space not to remove the calcareous algae. And so we do that underwater, and then out of the water, we need to set the cribs or ensembling them. Those cribs are made out of, they look like pools because we inflate them, so those are plastic base. And they have this setting structure or a base structure made out of PVC. So they and I'll tell you like the final, how it looks at the end. So we inflate them with air, as if we were just ensembling a pool. Also, we will attach this crate. So these are the crates that Mar, which is one of the leading NGOs doing this sexual reproduction out there in Dominican Republic, uses. So we'll have to prepare all this from advance before even going into monitoring and trying to collect gametes, just to be ready once we have our pool of gametes fertilized. We have everything already set on the water. So this is the previous process. And then once it's ready, it looks like this. It has like it looks like a little tent on the water, a little pool with a covering on the top. And so we need to locate those cribs in a site. So this year, and as many of you, if you were in Roatan, we had some talks or some webinar as well with once the sacred technicians came to Roatan and visited. And we were talking and also surveying the sites that we will locate these guys. So the crib will be located right in Half Moon Bay, right in West End. And I know there's going to be a lot of people, there's going to be a lot of curiosity drawn because of the cribs, but there's going to be in a lot of campaigns also trying to be done. And to avoid to be touched and try to educate people like what are we doing so we can leave them alone also, because we need to keep an eye as well, so we cannot leave them just further away or either really deep into the water. So we need to locate them at the site, anchor them, or simply attach them into an underground, because these are not permanent. These are only temporal attachments. There's only a crib that will stay for a month in the water. And then we will place those crates inside this crib with the substrates on them. So we will count the substrates to have specific numbers inside each crate. If we want to rear 1,000 of the substrates, so we will place, for instance, in this 10 or 9 crates that are inside, we'll place the exactly amount of crates or equivalent for each one. And this is how it looks from the top, but it's above the surface and below the surface of the water. And this is a picture from those cribs in Dominican Republic. And you can see like the PVC base is like gives this like structure, like a skeleton. And then inside there's the crates that are being held by the water, because everything is under the water. And the little cover like looks like a tent because it's only protecting because of the sun. So after we locate the crib, we assemble the crib, we locate that and to the site. Then, and when we place the substrates on them, what we'll do is like we'll be monitoring the crib and also the substrates, because we will have during the day, and let's say that during the night we monitor, collect gametes, we do the fertilization, and this happens into a single day, like the whole monitoring, collecting, fertilize them. Then we have the whole pool to put it back into our fertilization that we have in our coolers or our container. We bring it into the site where is our crib, and then we pour it down. We pour it in our cribs inside where there's this substrates, and then we leave them. What we were being recommended is that the less we touch it, the less we go into them, it's better. So we will be keeping maintenance, cleaning them once a week, because there will be a lot of algae also growing from the outside of the cribs, but we will still be like monitoring, keeping an eye on them. And also another way of monitoring the little larvae to see if they are perhaps settled is using blue light. So we will learn as well to count or even identify which is like coral larvae, because in between of this whole substrates, there's not going to be only corals growing on them, because it's on the water. We cannot control what is coming into them. So it's been research and has been really verified that under blue light the corals glow and they look amazingly, there's like fluorescent color. So this is a way that we also use for them to count them or study them and monitor them once they are in the substrates. So once they're in the substrates, there's a time as well that we need to wait for the larvae to settle down. If we put it today, let's say today during the night, we pour down all these gametes into the substrates, the next day they will still be floating around trying to look for a place for them to settle down. So there's a timing for each species. Some species takes up to 2 weeks, 1 week, or 3-week period. And during this week, we will keep the cribs in the water and providing this like monitoring and the monitoring of each substrate. So once we see that the larvae are settled down, there will be the outplanting time. And the outplanting time, it's like basically once we have finished everything and we need lots of hands trying to outplant them. We will decide, because there's like different techniques as well of outplanting them. If we want to monitor them, we want to know where we're planting them. So if you can see here in UNAM, which is the University of Mexico, they do this technique, and what they do is that they tag as well some coral babes or recruits, and the seeding units that they are placing, and they place in transects, like a line into a certain area where they know already, so they can be monitoring throughout the time, or throughout a month, a 6-month period, throughout a year. So it's not like a weekly monitoring, because corals, if we grow them from babies, it takes up for them to grow for a year, it takes them like a centimeter to grow. So planting them, also the substrates, as you can see, they are like a star shape. And one of the main objectives of that shape is because we can attach them in the reef, like straight into reef folds. So they provide as well like this an easily way not to use like much more equipment, but easily attach them to the reef. And we will be outplanting them. But also we will be monitoring outplants later on. And as you can see here in the pictures, some of the methods is benthing transects. So we lay down measured laces where we count down like the percentage of outplants, or the percentage of how many substrates that we lay down they have recruits in their basins, or the unit sits. And you can see here on the left, these are the recruits. So this is a picture that belongs to the seeker, and this is how this lace, like red lace that is around, it's a tag, and you can count 6 recruits that are different sizes. And I wanted to show you here one of the pillar corals that are from the recruit from Mar, Dominican Republic, from 2020, if I'm not mistaken. And this is estimated to be 1 centimeter. So it's a 1 year. So this is 2021, I believe. Not 2020, yes. So it has 1 year, almost 1 year and a half. And this is how it looks. It looks, to me, it's the most like amazing like baby coral I've ever seen. It's really rare to see pillar coral babies. And they also monitor. So this pillar coral, it's inside of one of those star-like substrates, and they keep it like hanging in some little tables. And they keep an eye, like, because there's a lot of efforts back in the history of this coral, like human efforts. So they're really like lovely but possessive with their baby corals. So they keep a really close eye into them until they believe that there's enough time for them to be independent. So yeah, so basically all that we would do is like the whole process that now you know that it takes up until the final objective, which is monitoring the one we have already outplanted. And just to mention a little bit, and what I've been repeating along the whole presentation, so this is one of the ways that we are trying to as well continue managing our outbreak, or SCTLD outbreak, that a lot of colonies that we have treated and we have tagged, those colonies that will be one of the from the ones that we will monitor, and probably that we can take and collect gametes and help them to reproduce, as well as the resilient colonies, because those resilient are the ones that we want to mix genetically or very genetically, so they can be more resilient to further diseases that we know that will come along. And as I strongly as well mentioned, that this will be helping to preserve reefs and recover populations of reef building corals. So this can help because we have been doing this through asexual methods, such as our coral nurseries. Now we can do it sexually as well with coral nappies, and we'll help out one way, because we're not stopping from doing either way. We cannot say like one way is better than the other yet, because both ways provide different or satisfy different needs that the reef needs. And this time, what we're trying to increase is genetic variability. And this way of doing it, it works. It's working a lot. Our mind will say that we would like to do like coral gardens as well. Grow corals to help endangered populations to recover, increase of this genetic resilience that will help them overcome outbreak diseases in the future, and provide protection to shore and habitat to marine wildlife. It means like a whole different factors that it keeps a healthy reef, try to help the reef to be healthy. So these are the ways that how we are going to, or how you can get involved, how we want to collaborate with everyone here at the Roatan and any other way, other parts of Honduras, and even further away from Honduras. We will create in this Ocean Youth Program, which we have secured some fundings, and this year we were granted as well to train a local community. And this local community includes divers, already like experienced divers, whether the ones that are being local here, but because they have stayed, that they come here and slowly stay here, and they have been working, collaborating with us with other projects. So we will train you guys, which would be, well, this is like the starting, this little webinar is like a small training as well. But also we want to train local island students, like young adults, because we consider that our corals and the future of our ecosystems are in the hands of the youth that we are now. So we want to provide them the capacities as well as the experience, so they can further have those experiences and knowledge, like base knowledge, so they can later on continue careers in this marine or science areas. We will also try to collaborate with intern students, intern university students, whether it be Hondurans and foreign countries as well, because we believe that the youth and inside of Honduras or here in the community are the ones that will maintain Roatans and Honduras coral reefs for the future. We want to continue work with local partners that we've been working since previous years throughout different conservation initiatives. So dive centers, the Roatans are really important because our monitorings are as well capacities being into every single diver, as well as dive owners, dive managers, working or helping in different ways are key for those projects to stay in long term. And just reminding that this project is a collaboration of 5 years with that we will be trying to rear these baby corals, but also this will continue like until its existence, until we create these coral gardens and help out the reef, which is really long term. And other ways that you can get involved is volunteering yourself as well. Is if you're a diver or if you're a snorkeler, you can volunteer as well in different stages of the sexual and coral restoration activities. As we saw, there's so many of them, and if you can't be in all of them, you can be present in any of those stages that your skills will help out as well. So you can volunteer and sponsor as well if you're a dive owner, dive manager. You can volunteer, sponsor time, boat tanks, because we have seen it as well as when we were doing monitorings and treating with acetyl-D, and we know how valuable this collaboration and connections are. And also another way is reporting sightings. because we have elaborated this calendar, we now will be sharing throughout our social media or throughout like close connections, possible timings. And so if you're an active diver and your instructor, if you're diving during these times, we will encourage you to provide also this valuable information. And as for now, if you can see in our social media, we've been already starting to post this about spawning seasons. And the first one that we would like to go for it is this brain coral, which is the groove brain coral. And we don't have much information on what's happening during the months of April, which is really less probable to spawn, but we would like to know in case that it does. So divers out there or snorkelers, if you see them spawning, or if you want to go and see what are they doing from the week from the 24th to the 30th, timing from 4:30 to 6:30, just keep an eye on it and have fun. And if you see anything, just snap pictures and take this valuable data as well. So there are different ways that we can get involved. And also additionally, it's an incredible underwater event that not only us as conservation NGOs or communities that we would like to develop this coral restoration projects, and as well as the manager protecting our area, but also there will be additional benefits, even as touristic opportunities in the future once we establish our calendars. And I know that who wouldn't love to see this, right? But this is like in the long term as well, once we have established all the base information, and also like structure what could be done in the future. So thank you, everyone. I hope it wasn't really loud, but I just wanted to like be very specific and slowly into transmitting or conveying all the information to you as clear as possible. So thank you, and Carolina, I reach out to you, see if there's any questions.
Thank you, Damaris. Okay, if anybody has questions about or doubts, you can let us know at the chat box, or Damaris will be happy to answer them. Actually, we have two questions. Somebody is asking, can people from mainland be part of the Ocean Youth Program?
Okay, so we will launch, there will be like several requirements. We will post these requirements. Basically, the requirements are that you need to, at least if you are from mainland, because we will be hosting this youth programs almost for more than 3 months. So you'll have to have somewhere to stay here. So basically that it will be a little bit more difficult for people from the mainland coming here. But if you have family, have like somewhere to stay, or to basically fund yourself and during that time, and if you can if you commit and are able to go into these requirements, which I believe it's like we would like for this youth go from like 15 years or 16 or 20 years, that range of age, as well as to be studying and yeah, just committed as well. Once we have all the requirements, we will post it and you'll see if you're able to go into the line as a youth program, because this program is basically, we would like to go into young adults that are in their last years of school, like go before university. And then for other ways of getting involved, maybe not the youth program, but as volunteering. That doesn't matter like how old you are or the timing, or if you're already a student or a mainland student, you can come as an intern or volunteering for more time. So there are different types, not only if you cannot go inside the requirements as being a youth program, there will be these other ways that you can get involved.
Okay, cool. Emma Doyle says, this is a great overview and excellent info and great use of photos. Thank you. This question that's coming to mind is about the level of investment required at the different stages, both setup and operations, capital needed, and in-kind support. Any comments that you might be able to give on that side of things will be interesting.
Absolutely. Yeah, and all those different stages, it's a high level of investment, yes. So I believe that we have several budgets already in around this activities, like because every state needs like different type of equipment, type of like physical investment or personal people being invested into this area. So I believe that from the beginning, since monitoring, and this is why we're trying to also get the whole community involved, because from going into monitor, we will need the time of the people, we will need tanks, we will need boats. So not everything as us as Roatan Marine Park, we will be able to provide to us. Let's say there's a lot of volunteers or not much volunteering, and we will be monitoring, let's say, these 10 priority species. And for this 10 priority species, we will need to monitor for 2 weeks in 2 months. So there will be a lot of exhaustion and to the volunteering or the staff that will be monitoring. So what we've been planning also is that we have different in different stations, different teams. So we can all together also, well, we can all learn everything until the final process, but as well in different stages, different teams, so we can have this energy is required, like human energy required to do since the morning or the previous days, for instance, cleaning, setting cribs all together, but monitoring these other species. Not for all the species, we'll go into the final stage, as I mentioned, and we will go only for one, but we can all like divide into this stages. In capital needed and in-kind support, and from in-kind support, that means like what we will be providing. We, as I mentioned earlier as well, we were granted, or we were able to be accepted for this grant, that we will have mostly the whole material will be covered by us. Also, there's other collaboration are providing us with the crib itself, with the materials to build the collectors, and some of other materials to like the mesh and weights and small things. We will be providing those as in-kind support. What else? Tanks as well. We were because we introduced this grant, so we will be able to fund a little bit with that, but mostly for this youth program. And also torches that we will be using. We have a few, but that's as in-kind, but as well as trying to encourage the other divers that may have them. So it's a little bit of what we have secured in funding, and basically a lot of the things that are mostly or highly evaluated as the capital needed will be provided by this collaboration with Seacore, which they provide first capacities, second the whole equipment to rear those larvae in the water, which are the cribs. But also what we've been having is that there's a lot of donations, so we can build as well like this little space that we can at least have our microscopes that we've been able to already enjoy or have them because of donations, because people probably cannot be underwater helping with their time and everything, but they do donate them. So there's a lot of like in-kind support as well coming, or that we already have. And yeah, so it's really interesting, yeah. And just thinking about it, it's a really, since the moment that we thought on, are we really like committing to do this. We knew that it was going to be a long and exhausting journey, but we believe that providing the capacities and going into the community and reaching out to others, like multiple hands will come along and will be trained, and we know for sure that this will be done by everyone. And this is like one of the things that we want also, why locals, we want them, because they are the ones that can stay here for a while, or if they leave, they know that they have here a place that their corals will be growing.
If somebody else have questions or comments, you can put them on the chat box as well.
And just to finish, everyone, so I mentioned that everyone can join in different ways. So if you have more questions as well, please send us an email or send us in any of our social media questions in regards of that. Because here, if you're already on Roatan, we'll be I'll be hosting other trainings, and maybe in May or June, in-person trainings, where we'll be also creating this collector net to have everything set when we have the whole equipment. So if you want to be part of that, just write down your contact so we can start creating group chats and following up with everyone.
If there aren't more questions, I want to thank Damaris for giving us this webinar on coral spawning and assisted sexual fertilization. If you want to know more about our coral spawning and how to volunteer, you can visit our website or our office at West End. You can also check out our Facebook and Instagram page, where we'll be posting more about this topic and our achievements on it. Thank you all for being here today, and have a good night.
Thank you, everyone. Thank you, Carolina.
Welcome.